our last discussion, we looked at some of the insights that you get from the one locus two allele model of viability selection, and our discussion was primarily on the theoretical insights that we got. And I promised that in the next discussion, which is this one, we are going to talk about some of the features of selection coming from experiments. However, I later realized that before we go there, it will be important for us to get a more real world sense through examples of some of the concepts that we discussed during those theoretical discussions. So, I am slightly changing the plan. In this discussion, we are going to look at some of the empirical examples of 2 or 3 of the concepts that we dealt with in the past 2 discussions. And the first thing for which we will look at an example of is what is known as overdominance. So, if you remember, overdominance is the situation when the heterozygotes have maximum fitness compared to both homozygotes. And the example I have chosen is very famous example, all textbook example. I am sure you have read about it in your class 10th or class 12th, wherever, and this is in the context of sickle cell anemia. So, you know that in our blood we have these red blood cells, and inside the red blood cells we have the protein called hemoglobin, which helps in transportation of oxygen. Now, in the hemoglobin there are multiple components of hemoglobin. One of them is called beta hemoglobin, and when it comes to the gene for the beta hemoglobin, there can be multiple forms. One of them is known as the S form. So, basically the way it differs from the regular form of hemoglobin is that it has a point mutation at one single nucleotide mutation, because of which the one amino acid changes, and because of the change in that particular amino acid, what happens is that individuals who are A, they end up being normal phenotype, whereas individuals who are homozygous for the mutant, they end up suffering from a disease called sickle cell anemia. Now, what exactly happens in sickle cell anemia? So, here what I am showing you are the red blood cells of humans, and these are the normal ones, the circular ones, and this one over here, this is the one which is an abnormal one. Okay, so you can see that it is looking like a sickle, right? Now, because of this, this particular cell is not able to carry as much oxygen as normal cells will be able to do, and because of that, the organisms or the individuals which are homozygous for the S allele, they end up having lots and lots of physiological complications, to the extent that most of them typically die by about age 5 or so. Okay, very few of them actually attain adulthood. So, this tells you that there is a very strong selective pressure against this allele, and therefore, you expect the frequency of this allele to be very, very low in the population. However, that is not entirely correct, and frankly speaking, the S allele is rather widespread in the globe. So, just to show you, the historical distribution of the sickle cell trait is over a lot of Africa, quite a bit of Middle East, and also India. So, obviously people ask the question that why is it that an allele which is so harmful in its homozygous form, why is it that it is so widespread? And as you know the answer is malaria. So, turns out that there is a very good correspondence between where you find the sickle cell allele and where you find malaria. And why is that? That is because individuals who are homozygous for SS, obviously they are in trouble, but individuals who are heterozygous for SS, for the S allele, that is basically the AS genotype, those individuals, they actually have an advantage. What is the advantage? Turns out that the malarial parasite is not able to invade the RBCs of these AS individuals, because of which these guys have a very good amount of protection against malaria. So, the way the whole thing is happening is that if you are a A genotype, then you are normal, but in places like these, you are going to have a very high rate of death because of malaria. On the other hand, if you are smallest individual, capitalist individual, you have sickle cell anemia, anyway your fitness is going to be very low. In these malaria prone areas, the heterozygotes actually end up having a much higher fitness because of the fact that they are being protected from malaria. So, this is a well known example. I will give you one more example which is again very interesting. This is related to this flower known as black vanilla orchid, Gymnadenia relicani. Okay, quite a tongue twister, but we will just call it the black vanilla orchid. So, turns out that this particular orchid, you can, or rather the flower of this orchid, you can find them in all kinds of shades, but three major shades are what I am showing you over here, so called black, so called red and so called white. Okay. Now, it is very interesting, why exactly do we have this? So, turns out that the red color morph, that is this one, this middle one, this one is heterozygous again for a single nucleotide polymorphism, which basically means change at only one base pair, which introduces a premature stop codon in a R2/R3 MYB transcription factor. Okay, does not matter what it is, some stuff in the genome. Because of this, it leads to a reduced expression of the anthocyanidin synthase gene. Okay, and this anthocyanidin synthase gene, this is what is responsible for a protein known as the red cyanidin pigment. So, the moment you have the mutation, the amount of red cyanidin pigment that is being produced goes down, and this is the interesting part. This, as far as the scientists can make out, does not affect any other phenotypic trait. So, those individuals which are so called black, they have both alleles normal, they produce lots of cyanidin, and therefore, the flower is extremely darkly red, we call it black. The ones which are heterozygous, they have one copy of the origin or normal allele, one copy of the mutated allele, therefore, their color level is somewhere in the middle, we call it red. And the ones which are homozygous for the mutation, they have both the alleles, so both the copies of the allele as the faulty one, and therefore, they produce the least amount of red cyanidin pigment, and therefore, it is very light color. Now, till this point, there is no difference between the three phenotypes except for the color. As I said, this does not affect any other phenotypic trait. However, what happens is in terms of the context in which these flowers are found. So, these flowers, they are pollinated by two major groups of pollinators, the flies and the bees. Now, turns out that these flies and the bees, they have very contrasting preferences for the flower colors. So, the flies, they prefer the white flowers over the black ones. So, that basically means they go more to the white flowers than to the black flowers. The bees, they do exactly the opposite, they prefer the black flowers over the white ones. Now, what about the reds? What about the heterozygotes? Turns out that in both cases, the heterozygotes, both pollinators have intermediate preference for the heterozygotes. Okay, but turns out that if you integrate over everything, then the red flowers, because they are intermediate for both of them, overall they end up getting relatively high number of visits. And because of this, if you actually calculate their fitness, which in this particular case was calculated in terms of how many seeds are being set, turns out that on an average it is the red flowers which have the highest number of seed sets, which is directly correlated with the pollinated visits. Now, because of this, the fitness of the heterozygotes is higher than both homozygotes, and as a result of this, you expect that in this population, this polymorphism between the two forms is going to be, between the two alleles is going to be maintained. Note, this is the important one. As I said, there is no difference between the two alleles except in terms of the color that they produce. They do not affect any other physiological feature of the orchids. However, the entire thing is being mediated by the preference of the insects, the particular preference of the two insects. Okay. Now, what will happen if all the insects, say all the flies, okay, all of them start preferring one color. For example, the example that I have taken over here is, let us assume that all the flies are killed, okay. So, you only have the bees. What will happen? As I already told you, the bees they prefer the black flowers over the white ones. So, now they are going to primarily go to the black ones, they will go to the red ones intermediately, and there will be very few visitations to the white. So, suddenly from a overdominant situation, this entire thing will flip. Now, the black flowers will have the highest fitness, the red flowers will have intermediate fitness, the white flowers will have very less fitness, and from a overdominance case, it will become one of those other cases that we were talking about, where you have the heterozygotes where you have a proper gradation in terms of fitness, homozygote 1, heterozygote, homozygote 2. If that happens, then what will happen? Then the allele which is leading to the black form in the homozygous form, that is going to be preferred, and therefore, the variation from the population will get lost. So, the basic point that I am trying to make over here are two. One, overdominance maintains allelic diversity in a population, we already saw that. The second point is that the entire stuff is context specific. Okay. What is the fitness of a type depends entirely on what context that genotype finds itself in. Under certain contexts, the system might be an overdominant one, under certain other contexts, it might have a very different kind of a fitness. Okay. So, after overdominance, we come to underdominance. What is underdominance? Remember, this is a scenario where the heterozygotes are less fit than both homozygotes. So, there are not too many well documented cases of underdominance in the literature. This is one of them. So, this is a butterfly, as you see, it is called the false wanderer, and you generally find it in Africa. So, now turns out that in this butterfly, there are two alleles, and each allele duplicates the appearance of a different local species of the butterfly. Okay. So, basically there are two different species, other species, not this one, not the false wanderer, and one allele makes it the false wanderer blue and makes it resemble one species, and the other allele makes this orange and makes the false wanderer resemble another species. Now, turns out that both these other species, both of them are toxic, which basically means that the predators normally leave them alone. Okay. So, this is a case of mimicry, where the false wanderer is mimicking other species, but it is mimicking two different species, and depending on the allele, it either mimics the blue species or it mimics the orange species. Now, what happens to the heterozygotes? Turns out that the heterozygotes have an intermediate appearance, and this intermediate appearance actually belongs to neither species, and therefore, the predators, they actually attack these heterozygotes at a very high rate, because of which the fitness of the heterozygotes go down, and it becomes a case of underdominance, where as I said, the fitness of the heterozygote is less than both homozygotes. Okay. So, that is about underdominance. The third thing that we looked at was frequency dependent selection. Now, if you remember, I told you that frequency dependence can be of many types, depends on how exactly the genotypic frequency and the genotypic fitnesses are related to each other. So, I will talk about two major cases of frequency dependence which have been studied empirically. One is called negative frequency dependence, and the other of course is called positive frequency dependence. So, in negative frequency dependence, the rare morph has an advantage. So, as the frequency of a genotype goes down, its fitness goes up, okay, negative correlation. So, again, this is something, there are quite a few examples of this in the literature. The one that I have chosen is in the context of these fishes known as Perissodis microlepsis. So, this fish, you find it in Lake Tanganyika in Africa. And the speciality of this fish, there are many specialities, but one major speciality of this fish is that this is a scale eating parasite. So, basically what it does is it just swims around, and then whenever there is a fish which is, let us say, not very attentive, it just goes and bites away a scale from one part of its body and then goes away. So, obviously the fishes from which it bites those scales, they do not really like this fish. Now, the other feature of this particular fish is that the mouth of this fish is not straight. The mouth of this fish is actually twisted, and turns out that in the population you have one set whose mouth are twisted to the left and another set whose mouth are twisted to the right. Okay. Now, what happens? So, obviously the one whose mouth is twisted towards the right. So, what I am showing you over here, this one, okay, this guy, it ends up attacking the prey, its prey from the left side, whereas this one, the other one where its mouth is twisted towards the left, it ends up attacking the prey from the right side, what I am showing you over here in this particular diagram. So, these are the preys. Now, turns out that when the frequency of any one of them, say let us say the right jawed parasite, when this frequency goes up, then what happens? Most of them are attacking from the left. If most of them are attacking from the left, the prey, this big fish over here, the one whose scale is being taken away, that prey is more attentive towards the left side. Okay. And therefore, when it becomes more attentive towards the left side, it becomes more and more difficult for these guys to snatch the scale, and at that point of time the fitness of this fish goes down. On the other hand, when this happens, then what happens? These guys, they get an advantage. Now, when they get an advantage, what happens? Their numbers go up, and when their numbers go up, opposite thing starts happening. The prey starts paying more attention towards the other side, the right hand side, and now it is the other morph which is at an advantage. And this sounds like a story, but this is not a story. People have actually shown this. So, turns out that they actually ended up looking at the relative frequency of these two morphs over time. So, here on the y axis they are showing the frequency of the left jawed individuals, and you can see that over time the frequency is just going up, down, up, down, up, down, exactly the way you predict that it should happen. Okay. So, this is a situation, negative frequency dependent selection, which ends up maintaining polymorphism in a population. Why is that so? That is because that each time the fitness of a genotype becomes high, the number of that genotype goes up, the number goes up, the frequency goes up, the frequency goes up, the fitness comes down, now the other guy's fitness is high, and this keeps on repeating itself, and therefore, you actually end up getting a polymorphism in the population in terms of the various traits. Now, this particular kind of selection, negative frequency dependent selection, is something that you need to keep in mind, because about 3-4 weeks down the line we will come back to this concept when we are discussing the evolution of sex. Okay. So, as I said, you have negative frequency dependent selection and you have positive frequency dependent selection. What is positive frequency dependence? When the common morph has a fitness advantage. Okay. So, just to give you an example, so what I am showing you over here are two species of butterflies. So, whatever is on top, this is known as Heliconius melpomene, these ones on the top, these ones, and what is at the bottom is another species called Heliconius erato. Now, for both species, it turns out that they are found all over South America, and although it is the same species, but in different areas of South America they look very, very different. So, if you just compare all the top guys, so this guy, this guy, this guy, they are all the same species. Okay. This one, this one, this one, they are all the same species, and yet you can see how different they look from each other based on where they are found. The same is true for the other species. However, this is very interesting. If you look at a particular species at a given area, there is hardly any variation. Okay, so all the variation is across areas, no variation within the area. So, what the hell is going on? So, turns out that both these kinds of butterflies, both the species, they are very distasteful to the predators. Okay. So, they have certain chemicals in their body which when the predators try to eat them, they really do not like it. However, to figure out what is going on, what scientists did was they took both kinds of species, but okay, we will deal with one species at a time. Okay. So, let us say we are talking about the second species, Heliconius erato. So, they took individuals of that species, marked them and then released them in a particular way. What is the way? So, for any given space they released two kinds of butterflies, one that was of the same species and resembled the local species markings, and the other which is the same species, but it is a marking from a completely different place. In other words, just to show you, so for example, suppose for this place they will introduce Heliconius the lower one is erato. They will introduce Heliconius erato which looks like this, and they will also take Heliconius erato, let us say from here, and they will introduce that over here. Okay, so two kinds of Heliconius eratos are being introduced in the same place. Similarly for this place, okay, local and from some other place. And when they did all that, they found to their amazement that always it is the introduced ones which have the local pattern which always survives better than the one with the foreign pattern. And why is that happening? That is happening because remember I told you that these are being predated upon by birds. Okay. Now, the predators, they when they are born, they do not have an innate ability to distinguish between what is edible and what is not edible. They learn over time that okay, if I eat this butterflies with this particular pattern, I am going to get a stomach ache, or it is going to be very bitter or something like that. So, turns out that these predators, they are not very smart, right, and they also tend to forget very soon. Therefore, in order for them to know, in order for them to learn, and in order for them to retain that, these guys, the prey has to be very common. If the prey is not common, then the predator will tend to forget that okay, I can't eat this kind of prey. Now, whenever you are introducing something from outside, as they did in this experiment, if those guys had the same pattern as the local one, then the predator is mistaking them from the for the common local pattern. However, the moment the predator sees something which has a different pattern, now the predator does not really know that this is the one that it does not want to eat, and therefore it tends to start killing it, and because their numbers are very less, it does not encounter them very often, and therefore it takes a much longer time to learn that I do not want to eat this particular type, particular pattern, and therefore these guys, the ones whose patterns do not match the local pattern, they end up facing a much greater mortality. They end up their fitness ends up going down drastically. So, this is a case where you have the common morph, the common phenotype getting an advantage and the rare phenotype getting a disadvantage. The same results were also obtained with the other species of butterflies also. So, that is why I said you concentrate on one species, the other species results are exactly the same. So, why is this interesting? Because in this particular case, this is operating exactly the opposite of the negative frequency dependent situation. Here, instead of maintaining polymorphism, positive frequency dependent selection will remove polymorphism from a population. Okay, so this is more or less what the examples that I wanted to share with you, and now in the next discussion we are going to go back to looking at some features of selection more from an experimental point of view. See you, bye.